Hello students, is the screen visible and am I audible?
Yes, ma'am. Good afternoon.
Yes,
Good
ma'am. Good
Good afternoon,
afternoon.
madam. Yes, madam.
Good afternoon. So let's begin with the today's session. So welcome everyone to the NPTEL course on novel technologies for food processing and shelf life extension. And the course instructor is Professor H.N. Mishra, sir, from the Department of Agriculture and Food Engineering, IIT Kharagpur. I am Shikha Sharma and I am a research scholar in the Department of Chemical Engineering, IIT Guwahati. So today's live session will be regarding week 9 topics, which includes respiration and ripening, modified atmosphere storage or modified storage packaging, active packaging technology, edible coating technology, and multipurpose CA controlled atmosphere or MA, which is modified atmosphere storage units. So for each of these topics, uh, for each there is one video, and in total there are total 5 videos regarding week 9 topics. So I hope you have already gone through that. And in this live session, we will be mainly like practicing the problems related to these topics. So if you have any doubt in between, or if you do not understand any questions while solving, please feel free to ask. So let's begin with today's session. So today's first question is regarding maturity of the fruits. So the question first says, which among the following instruments can be used for the determination of the maturity of fruits? So is it like rheometer, Rancimet, Instron Universal Testing Machine, or Kramer Shear Cell? So maturity of fruits can be determined using which property? And for calculating that property, which instrument do we need? Anyone? Which properties we determine while understanding like the maturity of fruits or the ripening of fruits? So can anyone please tell me what are the properties name? So we understand like sometimes when we try to understand whether the fruit has been matured or not. So what we calculate weight loss, right? So what is the weight loss during storage? One property is this. So what we usually do, we see like what happens if there is any fruit. So while storage, its weight will constantly, it will like with some particular rate, it will decrease with the time because there is a water loss, right? So what happens in the fruit? There are the fruit cells or the plant cells. In that plant cell, there is like this is a plant cell. So there is water inside this plant cell. So what happens because the climatic fruits, especially in the climatic fruits. So climatic fruits, they after we harvest them or the after post like in the during the storage or in the post harvest time, they continue to respire and transpire. So that means in the mainly in the climatic fruit, we talk about climatic fruits, right? So in the climatic fruits, this water inside the plant cell, it will form like it will evaporate and it will go out in the environment. So the water is lost inside the plant cell. That means water is losing. That means moisture is losing or weight is losing at a constant or at a particular rate during the storage. One parameter is moisture loss or the weight loss. Second parameter because of the process of transpiration, which is evaporation of water, and that's why the turgor pressure inside the uh, cell it reduces, right? Second is the firmness or the texture. Firmness or texture. How when we are eating any fruit, if we see initially it is very hard to chew, and after particular time of storage, it will become soft. It will not be that hard as before as on the first day, right? So because why this firmness is decreasing or the fruit is softening, because there are different enzymes like pectin methyl esterase and polygalacturonase. Such enzymes are there which which like pectin methyl esterase, what it will do, it will break the pectin, like in the pectin polymer, it will actually act with the methyl ester groups, and like there is a particular reaction. So because of that, the pectin molecule will start, like the integration will lose. So cell integrity or the cell wall integrity will become loose, and it will become, the fruit will become softer, or the firmness will decrease as we store the fruit for longer duration of time. So the second is firmness. Another parameter is color. So we can notice that there is a color change in the fruits while we store a fruit, like in the apple, if we see, if we put cut apple, so there will be browning of the cut apple. So because of the different enzymatic browning reaction, so this the color also changes sometimes. So there are such different processes happen when also the respiration happens. So there will be more production of the, like in the head space where the fruit will be there. The CO2 production will be there from the uh, fruit. So we can measure the CO2 if the CO2 production increases in the air, so that means that the fruit is undergoing ripening process. Another is ethylene production. If the fruit is undergoing the ripening process, then it means that there is more of ethylene production. So there are different properties we can measure, and we can understand that because of these properties, there are different properties. If it goes beyond this level, these property like weight loss, if it goes beyond maybe 5% or maybe 10%, so the maturity of the fruit is happening, like that we can see. Also there are different biochemical processes like uh, conversion of the complex sugars, starches into the simpler sugars like glucose, fructose. So we can measure the amount of the simpler sugar in the fruit, and we can understand that if the simpler sugar amount is higher, that means that more is the maturity. So there are such properties we can measure from different instruments, and we can understand that maturity of a fruit is happening. So rheometer is the equipment which measures mainly viscosity, and there are different, like, so option C is correct here. That is the instrument which determines maturity of a fruit. So Instron universal testing machine is the one here, which determines maturity of the fruit. Basically, this equipment is used to measure the texture of the product, which is the equivalent to firmness property it is going to measure. So, rheometer, first option is not correct, because rheometer is the equipment which is used to measure the viscosity of any solution, flowing or liquid solution. Viscosity we measure usually from the rheometer. So rheometer cannot be correct here, because it measures viscosity measurement and consistency measurement. Then second is the Rancimet. Rancimet basically measures oxidative rancidity of the oils. So this is the different rheometers we usually see, and power rheometers. And then this is the setup of the Rancimet. We discussed this in previous lectures, that Rancimet is, in the Rancimet method, what happens, air is bubbled through the oil, and the increase in electrical conductivity we measure, because it changes due to the generation of the oxidation products, usually at the 100 degrees Celsius, and we known as this, the induction time is the parameter which we get from the Rancimet method. So from here air will go, and here sample is there, and there will we will going to measure conductivity of the cell. By that, because if there is a production of oxidative production, more oxidative products will be there, then there will be change in the electrical conductivity. By that we can understand that oil is rancid or is undergoing the oxidative rancidity or not. So this equipment also we cannot measure maturity of fruits. So third is Instron Universal Testing Machine. So which is this machine? So in this what happens, texture changes represents the maturity of the fruit. As we discussed, that the texture it changes, that means the firmness, the firmness is either increasing or decreasing. Usually the firmness decreases with the storage time, so we measure from this machine. So determination of the maturity involves instrumentation such as the Instron Universal Testing Machine, which can be equipped to perform texture analysis, including the attachments of Kramer shear cells or puncture probe measurement. So sometimes what we do is, this is the fruit, so there will be one probe which will come and puncture the fruit, and the force which is required to puncture the fruit will be measured as the reading, which tells that how much is the fruit is firm. If less force is required, that means fruit is soft. If more force is required, that means fruit is hard. Then there is also compression test, another method. In that what happens, probe will come, touch the surface of the fruit and will go. So it will just, it will not puncture, it will just compress and understand the force, the back force, which is like if we apply some force to fruit, so fruit will going to apply some backward force to the probe. So it measures, in the compression test we measures the force which is applied by the fruit in the response of the force which is applied by the probe. Then it is used to test the tensile strength or the compressive strength. Then this equipment also is used to measure fractural strength, bending, shear, hardness, torsion, etc. But all these are usually more relevant in the kind of like in the mechanical system, mechanical engineering system, where we want to measure the stiffness of any composite material. But here in the fluid technology we need to measure the chewiness, hardness, springiness, cohesiveness, such terms from the universal testing machine or the texture analyzer. Then is this picture of in the left hand side is of the Kramer's shear cell, which measures fruit texture that simulates biting by using a moving probe with blades to shear, compress, and extrude a sample, providing data on the force and the displacement required for these actions. So this also measures fruit texture, but it tells more about the shear, compression, or the extrude. We extrude the sample and we get the force and displacement uh, like the graph between force and displacement. And this is by this method we measure the fruit texture or food texture, any food texture. And so this option C is correct, which is mainly used for the uh, texture analysis or the firmness by using the two methods, either the compression method or the puncturing the fruit and measuring. So it measures on the two method. Is it clear, this question?
Yes, ma'am.
Okay, so can we move to next question?
Yes, ma'am.
Yes, ma'am.
Okay, so we will be solving this question. So please, can you like use some pen and notebook? How much? Okay. So this question is a numerical problem where 0.3 mm thick cellophane wrapping packaging material is used and its area is 0.2 meter square, was used for packaging. Then the vapor pressure inside and outside the package is 50 and 5 mm Hg respectively. The water vapor permeability of cellophane is 1.82 into 10 to the power minus 10 meter cube per second meter square atm pressure per meter. So we need to calculate two things. First is the diffusion flux and second is the loss of water vapor in gram per day at steady state. We are assuming that water vapor loss is in the steady state at the steady state condition. So first is the what is water vapor permeability. So it is the time rate of water vapor transmission through a unit area of a flat material of unit thickness induced by unit vapor pressure difference between two specific surfaces under specified temperature and humidity condition. Water vapor permeability is measured by the standard ASTM method, ASTM E96, and which follows the gravimetry method. So let's solve first part. So it is given in the question, that is the thickness of the film is given as 0.3 mm. I'm so sorry. This is 0.3 mm here, 0.3 mm, right? Equals to 0.3 into 10 to the power minus 3 meter. So we can convert it into meter. Then is the surface area of the film is given as 0.2 meter square. Water vapor inside the package inside is 50 mm Hg. So 50 mm of Hg. So if we want to convert into the ATM, so 1 ATM is equals to 760 mm Hg. So that means 1 mm of Hg is equals to 1 upon 760 ATM, right? So if in 1 mm of Hg, 1 upon 60, 1 upon 760 ATM is there. So in 50 mm of Hg, it will be 1 upon 760 into 50. So that is equals to 0.0657 atm. Then vapor pressure in the outside of the packaging, which is given in outside in 5 mm Hg, right? So same we can convert it into 5 mm of Hg into atm, which is 1 upon 760 into 5. So it is going to be equal to 0.00657 atm. Then permeability of the packaging is given as in the question 1.82 into 10 to the power minus 10, right? So it is this, and then loss of water vapor and diffusion flux we need to calculate, right? So two things we need to calculate right here. So we know that according to the Fick's law of diffusion, that flux is equals to diffusion coefficient into concentration gradient upon thickness of the material. This we know from Fick's law, right? So from the diffusion coefficient, in the first part we need to calculate diffusion flux. So diffusion flux is equals to the concentration change, which is the change in the, also we can write it as change in the pressure, right, into the permeability coefficient, which is given in the question as this, right? And then the thickness is Z2 minus Z1, thickness of the material, and since we are using here pressure and not the concentration term, so we will divide with the molar volume. We will convert it into uh, the pressure into the volume. So diffusion flux is equals to, first is the this is permeability, water vapor permeability, which is this, right? 1.82 into 10 to the power minus 10. So 1.82, this is the permeability, 1.82 into 10 to the power minus 10. Then change in the pressure. So from inside to the outside of the packaging material, what is the change in the pressure? So it is, we converted into atm, which is 50 is inside, which is PA1. So 0.0657 here we calculated, right? So 0.0657 minus 5 mm of Hg, which is equivalent to 0.00657 atm, divide by 22.414, since molar volume of a ideal gas at standard pressure and the temperature is 22.414. So it is 22.414 into thickness, which is 0.3 mm or 0.3 into 10 to the power minus 3. So this is the thickness. Now if we can you please mute? Please, can you mute? I'm so sorry. This is so noisy. Thank you so much. Yeah. So if we minus 0.0657 minus 0.00657, so this minus is 0.05913, this value, right? Into 1.82 into 10 to the power minus 10, which is this. Again, this is my voice is repeating. Yeah. Please, can you mute? Thank you so much. So, please everyone mute yourself so that I'll not get disturbance. मैम वीक 9 का क्वेश्चन काफी टफ है. So we are solving, right? Maybe that will help you. Okay, so we were solving this question. So 1.82 into 10 to the power minus 10 into, since we subtracted and we got 0.05913, divide by, when we multiply this value 22.414 into 0.3, it will going to give 6.7242 into 10 to the power minus 3. So when we multiply 1.82, this value, into this value 0.05913, so we will going to get 0.1076, then divide by 6.7242, the same value, then it is 10 to the power minus 7 minus 10 and here minus 3, so the result will be 10 to the power minus 10 plus 3, which is 10 to the power minus 7. So when we divide this value and solve, we will going to get diffusion flux as 1.6 into 10 to the power minus 9. So option D is correct in the first case, which is about diffusion flux. So diffusion flux is 1.6 into 10 to the power minus 9. So this part is clear. So we can we move to part B, which is about loss of water.
Yes, ma'am.
Okay.
Yes, ma'am.
Thank you. So the second question is about loss of water vapor, gram per day. In gram per day, we need to calculate. So the amount of water vapor is diffusion flux into surface area into time. So diffusion flux is, we calculated just now, 1.6 into 10 to the power minus 9 into area into time. So here is the 1.6 into 10 to the power minus 9, same, into 18 is the, like, we, since it is in the moles, this value is in kg moles, right? So to convert it into the, because we need to convert in grams per day, so grams is equals to number of moles, which is 1.6 into 10 to the power minus 9 into molar mass, which is 18, because this is water vapor, so water molar mass is 18. So when we multiply number of moles into molar mass, this whole value we can get in grams, right? Into, so this is given surface area is 0.2. So 0.2, and then since and this is the time, this value is for the time, so we need to calculate in days. So for 16 to 60, 60 seconds into 60 minutes into 24 hours in one day. So 16 to 60, 360 into 24, and this 1000 is for, because we are because there is, like, this is kg mole, so we need to convert into grams. So multiply with 1000. So when we solve this, the loss of the water vapor will be 0.497 grams per day. So C option is correct here. Is it clear, this question, to all of you?
Yes, ma'am.
Okay, thank you. Let's move to the third question, which is about respiration question. So in column 1, there are 4 RQ values, and we need to tell in the column 2 that the related metabolic substrate or the process. So would anyone would like to share his or her opinion on this question?
Option C.
So we got one as
C.
option C.
C, ma'am. Yes.
So everyone agreeing to option C?
Yes, ma'am.
Yes, ma'am.
Yes, ma'am.
Yes, ma'am.
Okay, so yes, all of you are correct. Option C is correct. Since we know that ratio of CO2 produced and to the oxygen consumed is the respiratory question. So RQ is RCO2 divided by RO2. And if RQ value, it is normally assumed to be equal to 1 if metabolic substrates are carbohydrates, and if substrates are lipids, then it is always lower than unity, because the ratio between C and O in lipids is lower than the ratio in carbohydrates. And if substrate is an acid, then RQ value is greater than unity. RQ is much greater than 1 when there is anaerobic respiration takes place. So that means the fermentation process is going on. So option D is correct. So everyone is sure about this. So we are moving to next question. So which among the following gases can be used to replace N2 from wine bottleneck before corking? Oxygen, CO2, argon, or SO2. So what happens in the corking of the wine? So wine, when it is get contaminated by TCA, which is trichloroanisole, which is this molecule. So this chemical compound, which is caused, like, because there is a mold or fungi present in the corks or the wood, because of that it get the wine get contaminated and produce like contaminated, and there is a corking of the wine, which is an unpleasant. So unpleasantly it alters a wine's flavor or the scent, aromas and the flavors, to the point that it becomes like it cannot be drunk before, because of a strong cork taste. So it is an undesirable change in the wine, which producers do not want, since it makes wine undesirable because of the strong unpleasant aroma. So uh, to prevent that, like, we need to like stop the oxidation process, and we need to replace the gases. So which among the following gases can be used to replace nitrogen gas from the wine bottle neck before corking?
Option B, CO2.
CO2. Anyone else?
CO2, ma'am.
Okay, CO2. Answer is D, C, argon. Because what happens during aging, if a wine is not protected from both the microbial spoilage and the oxygen, then it may spoil the wine. Protecting wine usually involves maintaining proper SO2 levels, which protect wine from the microbial damage or the fungal or the mold damage, and keeping containers full is the second option. Why we keep containers full? So that there is no oxygen in the headspace gas. Additionally, purging your headspace with the inert gases to effectively remove oxygen. So we purge the neck of the wine bottle. This is wine bottle. If so, there is the neck portion, headspace portion. So there we remove oxygen, so that we can protect wine. So because for that we use noble gases like argon and the xenon, and we modify atmosphere in the wine headspace. So argon can replace nitrogen to fill in the wine bottleneck before corking. So option C is correct because it is a noble gas. Is this option clear?
Yes, ma'am.
Okay, so it's not CO2, it's argon. So next question is about N2 generator in a CA or MA chamber. Which among the following is the operating principle of N2 generator of CA or MA chamber? So option A is saying slower kinetic diffusion of oxygen molecule into the pore structure of carbon molecular sieve. Then faster kinetic diffusion of, here is oxygen in the second, nitrogen molecule in the pore structure of the carbon molecular sieve. Third is faster kinetic diffusion of oxygen molecule into pore structure of the carbon molecular sieve. And option D is faster molecular adsorption. Here it's adsorption, not diffusion, of N2 molecules in pore structure of carbon molecular sieve. It's from the
Option
last
D.
topic. Option,
Option D, madam.
okay, option D.
Option C.
Option C. So let's see. It's option C, which is faster kinetic diffusion of oxygen molecule into the pore structure of carbon molecular sieve. So what happens in the nitrogen generator? What happens? Nitrogen is separated from the air mixture in the nitrogen generator. The separation of nitrogen takes place in an absorber vessel filled with carbon molecular sieves. So there will be carbon molecular sieves and nitrogen will be separated from the mixture of gases in the nitrogen generator. The operating principle of nitrogen generator of controlled atmosphere and modified atmosphere chamber is the separation of the nitrogen from rest of the air, which takes place because of the faster kinetic diffusion of oxygen molecules into the core structure of CMS, which is carbon molecular sieve, then N2 molecules, because the oxygen molecular size is smaller than the nitrogen molecular size. So CMS absorb oxygen, CO2 or the moisture. So it will absorb other components in the air mixture, in the compressed air mixture in a short period of time, and compressed nitrogen is available as an output. So it separates nitrogen like this method. When the pressure decreases to the atmospheric or vacuum levels, the CMS, which has adsorbed oxygen gas and other gases, easily dissolves them and it is regenerated. It can again reuse then, because when we change the pressure, the oxygen or the other gas which is entrapped in the carbon molecular sieves, it will go out and the CMS can be again used. So adsorption and regeneration process are done alternatively between two adsorption columns, and nitrogen gas can be made available continuously from the air. So this first diagram on the left hand side is about nitrogen chamber, where here air is going in column here first and here also, and then here are CMS, and in last we will get nitrogen as a pure product. So this is on the right side, there is a diagram of CMS where this is the magnified structure, where since this in the carbon molecular sieve there are different pores like the mesopores or the micropores, where of different of the mesopores and micropores are the one which has different dimensions. So in the pores we can understand that in the carbon molecular sieve what happens, there will be different pores. So if because oxygen molecular size is small, so it can go and accumulate in this pores, whereas nitrogen molecular size is bigger, so it will be outside. So so other components of the air mixture will be inside the sieve, and other which is the bigger molecular nitrogen, it will be outside and separated out, and we can collect from here, we can collect nitrogen. So carbon molecular sieve, when CMS absorbs impure gases, what happens? Macropores and the mesopores only function as channels, and the adsorbed molecules are transported into the micropores. So CMS side contains a large number of micropores, which enables molecules with smaller kinetic size to diffuse rapidly into the pores while restricting the entry of larger diameter molecules. Due to the different interdiffusion rates of gas molecules because of the different sizes, the component of the gas mixture can be effectively separated. Therefore, in the production of the carbon molecular sieve, the pore distribution inside should be in the range of 0.28 to 0.38 nanometer according to the molecular size. Within the pore size range, oxygen molecule can quickly diffuse into the pores through the micropores, while nitrogen molecule is having large size, so difficult to pass through the micropores to achieve the separation of oxygen and nitrogen. So the pore size is basically the main principle or main basis on which the carbon molecular sieve separates oxygen and the nitrogen. If pore size is very big, then what will happen? All the gases like nitrogen, oxygen, everyone will going to go in this pore. And if the molecular size, so if the pore size is very small, then neither of the oxygen nor nitrogen, none of the gas component can go inside the pore. So there should be optimal size of the carbon molecular sieve to separate oxygen and nitrogen. So option C is correct, that there should be a faster kinetic diffusion of oxygen molecule into the pore structure in the CMS. Is it clear, this question?
Yes, ma'am.
Thank you. Can we move to next question?
Yes, ma'am.
So it is also a numerical question. A 2 mm thick plastic packaging which is made up of LDPE has a surface area of 400 centimeter square. Partial pressure difference of oxygen across the film is 0.21 atm and permeability coefficient is 4.18 into 10 to the power minus 8. We need to calculate rate of oxygen transfer inside the packaging material. So anyone, please try to solve this question, or and please let me know which formula we should apply here for calculating permeability coefficient. So let's solve this question. Thickness of the film is 2 mm, which is given in the question, 2 mm. So we, since here mostly we use centimeter, so we, instead of converting into meter, we are converting into the centimeter because maximum values are in centimeter. So 2 mm is equals to 0.2 centimeter. Surface area is given as 400 centimeter square. Partial pressure difference is given as 0.21 atm. Permeability coefficient of package is given as 4.18 into 10 to the power minus 8. And we need to calculate rate of oxygen transfer. So the permeability coefficient formula is, permeability coefficient is equals to, this is given in the slide, this formula. So is equals to volume into thickness divide by area into time into difference in partial pressure. So in the lecture video, it is already discussed this formula about permeability coefficient. So we are going to apply this formula here. So we need to calculate rate of oxygen transfer. So rate of oxygen transfer, that means volume, this term, rate, and which is volume of the gas or volume of oxygen, and rate, so divide by time. If there is rate term, that means that is divided by time. So volume divided by time is equals to permeability coefficient. We are just rearranging this formula into area, this value, into difference in partial pressure P delta P divided by this thickness value. Okay, so we will going to get this formula. Now we just need to apply this formula equals to 4 point, because it is already given in the question. This permeability coefficient of the package is 4.18 into 10 to the power minus 8. So 4.18 into 10 to the power minus 8 into area is 400 centimeter square as here, into partial pressure difference. So it is also given as 0.21 atm divided by thickness, which is 0.2 centimeter, right? So here we used the same value. When we solve, like we multiply 4.18 into 400 into 0.21, then we will going to get 351.12 into 10 to the power minus 8, and in the denominator its thickness 0.2 centimeter. So rate of oxygen transfer, when we solve this part, we will going to get 1.75 into 10 to the power minus 5 centimeter cube per second. So option A is correct. Is this question clear to everyone?
Yes, ma'am.
Okay,
Yes, ma'am.
clear to all, right? So let's move to next question. An active thin blank layer containing blank is applied inside the packaging material for removal of limonene. So we usually apply some layer into the packaging material so that we can remove limonene. In the usually it is found in the fruits like citrus fruits or maybe grapes, mainly citrus fruits.
Option C, ma'am.
Option
Sorry.
C,
B.
B.
B, cellulose
C.
acetate and limonene.
इसमें बस ऐसे वीडियो में
Option B, ma'am.
Okay. Option B
C.
and option C. So can you both, can you give your explanation how you are selecting like B and C?
Enzyme has to be naringenes, but I'm confused between cellulose, nitrate, and vinyl acetate.
Okay.
Naringenes is a non-bittering compound, right? So it might be limonene.
Okay.
Maybe. But it has to be naringenes. I don't remember exactly. I haven't studied limonades.
Actually, uh, option B is correct, because usually this we have, because we need to remove Limonene, right? So the flavor and odor scavengers has been utilized for debittering of citrus fruits, juices, or sometimes grape juices. The cause of bittering compounds in the grape juice or in other citrus fruits, mostly like in the grapefruit, it is naringin, which is a glycosidic flavonol, and limonene, which is triterpenoid lactone. And if it's this limonene concentration, if it goes beyond 6 ppm, then there will be a bitter taste in the juice. Limonene is formed as a result of heat treatment of the juice during processing and chemical action in acidic medium. So what happens? This reaction happens in which during the processing this LARL, which is limonate A ring lactone, it is converted into limonene, which is a non-bitter compound actually. So it is a precursor substance of the limonene in citrus fruits, and it will be released into the acidic medium when like the juice is having acidic medium. So this compound LARL, it lactonized and converted into the limonene under catalysis of limonene D R ring lactone hydrolase. This enzyme, because of this enzyme, or we can say this is limonenase enzyme. Because of that, like the compound LARL, it converts into limonene. So we use usually thin cellulose acetate layer, which is applied inside active packaging or the packaging material for the removal of limonene compound. This layer also, like, it consists of fungal derived enzymes, which is naringinase, and it hydrolyzes naringin also to these two compounds. First, naringin is converted into uh, prunin, and then and this raminose, and further uh, because of the help of beta D glucosidase, it converts into narangenin and D-glucose. Two compounds will be get. Both are non-bitter compounds. Also, naranginase alkyl groups adsorb limonene. So, because of this reaction, this cellulose acetate is basically used for the layering in the packaging material to remove limonene. Is it clear?
Yes, ma'am.
Yes, ma'am.
Okay.
Yes.
Thank you. So we are moving to next question. Dash is the process of coating where the product to be coated is deposited into a large rotating bowl where the coating solution is then ladled or sprayed. Dipping, spraying, fluidized bed, and panning.
D. Panning.
Panning, right?
D.
Panning D.
Yes, panning is the correct answer. So what happens in dipping? Anyone, please?
We have to dip that substance that to be coated in a solution.
Correct. And then spraying. Other people, please. Everyone will tell one each. Okay, so she has already answered dipping. Now other people spraying. I want someone else to tell what is spraying.
The chemical is sprayed with nozzle.
Chemical or the coating solution will be sprayed through
Nozzle.
nozzle, the sprayer spraying bottle, right? And
Yes,
then
ma'am.
is fluidized bed. So what happens in fluidized bed? Some boy, I want some boy to answer this question because two females already answered, and now I want some boy to give answer. Anyone? What is fluidized bed? What happens in fluidized bed? Please unmute yourself and answer.
Ma'am, product is in motion in this. Like air is passed from the bottom, made the powder product which has to be coated is in motion, like, and
Oh,
the coating solution is sprayed on top, so it
okay.
ensures uniform mixing.
How fluidized bed term comes? A process, Anuj, can you please speak? A process equipment used extensively to reduce the moisture content of the fluid. Moisture content of the fluid. We are talking in terms, okay, we are talking actually in terms of coating, how coating is being applied using fluidized bed. And what is the, because she has already answered, so any, we just need to add some more points, like what is, why it is called as fluidized bed, because there should be some bed is forming, something is happening, right? Please, anyone compliment her answer. Because of air and there is some density of the product, so we will give some pressure of the air which just make a fluidized bed of the particles. Like we'll calculate first the density of the product or the mass of the product, so we will apply that much, only that much of the pressure of the air, so that it will make flu like a, it will the product will continue travel and there will be a bed of bed is formed, which is of of like that, from the downside there will be some amount of the pressure of the pressure of the air will come, and that will make a bed fluidized. That's why the term is known as fluidized bed. So let's move to the panning. So pan coating process consists of depositing the product to be coated onto a large rotating bowl referred to as pan. So here is the product in the bowl, which is a pan. The coating solution is then ladled or sprayed into the rotating pan. So this is air atomizing nozzle. From this coating solution will come and coat the product. The product is tumbled within the pan to evenly distribute coating solution over the surface of food material. So this will continuously in the rotating, this pan will continuously rotate for uniform coating, and the forced air, either ambient or at elevated temperature, is applied to dry the coating. So here there will be exhaust gas which will dry the coating material. So this is panning, option D. Next is the numerical problem. So let's solve this question together. A packaged food material of the food product has a shelf life of 12 months. If the package thickness is decreased by 20% and its surface area is decreased by 30%, then what will be the new shelf life of the food material in months? So let's solve this question. Shelf life of the food is given as 12 months. Already the shelf life is 12 months. Now we are changing in the packaging material. So initial thickness we are considering x1, and then it is we are decreasing x2. The second thickness is x2. Surface area is changing from A1 to A2. So the new shelf life of the product will be T2. So T2 months it will be the shelf life. We have just assumed. Now from the permeability coefficient formula, which we just now discussed, that permeability coefficient is equals to volume into thickness divided by area into time into difference in partial pressure. So from here we can see time, this term, thickness, this term, and area, this term. So if we shift time here, so time is directly proportional to thickness and inversely proportional to area. This relation we can get. Now, since we are changing thickness and area, so when there is a T1, which is the shelf life, initial shelf life, so T1 is equals to x1 by A1, which is initial thickness divided by initial area, divided by T2, which is after changing the thickness of the packaging material, what will be the shelf life, is equals to x2, which is new thickness, divided by A2, which is new area. So x1 by A1 will be same, and since in the question it is saying that the packaging thickness is decreased by 20%. So if we are having 1 as packaging initial packaging thickness, so it will be 1 minus 20 by 100, because 20% is decreasing from the initial, which is 1. So 1 minus, this is 0.2, 1 minus 0.2 will be 0.8. So initial thickness will be 0.8 x1, sorry, final thickness will be 0.1 x1, and then area is decreased by 30%. So that means 1, or we can say x1, which is from initial, sorry, A1, I'm really sorry, this is A1. So from the initial thick surface area, we are decreasing around 30%. 30% we are decreasing. So that means it is 0.3. So 1 minus 0.3 is 0.7 of A1 is left. So area is 0.7 A1. So x1 cross A1 cross. So T1 by T2 is equals to 0.7 divided by 0.8, which is 0.875. So since we need to calculate T2, which is the after changing what is the shelf life, is equals to T1 by 0.875. So T1 is given as 12 months. So divide by 0.875 is equals to 13.71 months. So answer B is correct here. Is this question clear?
Yes, ma'am.
Clear to everyone?
Yes. Ma'am, I have a question.
Yes.
Ma'am, if we will not do numerical questions in final exam, will we pass through theoretical portion?
So, since there is no weightage fixed as such that this much is should be numerical and this much should be theoretical, so you must do, like, there may be more numericals and less theory, or vice versa, like more theory and less numericals. And numerical is just not in this part, it's there in entire syllabus, right? So it's very, very important or crucial to solve numericals and to understand, and like otherwise it will be difficult, I think.
Okay, ma'am.
So at least basic numericals uh, you should attempt. And if you solve the assignments and you are solving here the numericals, so I don't think there will be much problem. So you
Okay,
try
ma'am.
to practice all these numericals. Yeah. Okay, so can we move to next question? So which among the following uses probes to replace air with desired gas mixture? So option A is about horizontal foam fill seal machines for rigid and semi-rigid packages, horizontal foam fill seal machines for flexible pillow pack pouches. Third is preformed container machines, and D is snor snorkel machines.
Option A, ma'am.
Option A, horizontal form fill machines for rigid and semi-rigid packages. Other people, please share your opinion.
Option A, madam, correct.
Option A, but answer D is correct. Let's understand why A is not correct and D is correct. So this kind of setup is a, yeah, snorkel machines. So snorkel machines do not need chamber presence during atmospheric modification. A large flexible pouch or bag is basically used where the food stuff is being kept, and there will be probes, or we know as snorkels, are entered into the pouches. So here in the pouch we have food, and these snorkels, they'll come inside the pouch and remove air, after which vacuum is broken by addition. So all the air will be removed and vacuum will form, and after that there we can add desired gas mixture in the pouch. So these probes are then removed and package is heated and then sealed. This machine is used for bulk packaging and for so-called master packs, in which individual retail packages are packed in a large MAP pouch or bag. So this is the airflow system. From this, air will come, and at the end, when we change the gas, the pouch will be heated and sealed. Whereas in form fill seal machines, they can make pouches also, and they can make thermoform trays also. So in the pouches, we can have vertical and horizontal machines both. And in the pouches, also like in the trays they make, and they will further heat seal the lids and make complete sealed pouches or the trays. in pouches, desired gas mix is introduced into the package into a continuous counter current flow to force out air. So gas will go and the existing air it will be forced out, which ends of uh, web are then heat sealed and packages cut from one another. In tray, products is being kept on the tray and then a vacuum is drawn, and then after that desired gas mixture is being introduced, and then there will be film of packaging material which is heat sealed to the base tray. Horizontal FSS, it is basically used for rigid and semi-rigid packages from deep drawn machine, and vertical and horizontal FFS is used for flexible pillow pack pouches from flow pack machines. Like this is a setup of vertical FFS machine, and the next the last third option was preformed container machine. So in the preformed container machine there will be preformed containers like this. These are known as preformed. So these are basically used to make PET or bottles, in which what happens, these preforms are being put in the blow molding machine and to make bottles. So preformed container machines work by taking preformed containers, test tube shaped objects which are threaded at the neck. So this is the bottles thread, and this is the where we blow gas to make into the shape of bottles, and transforming them into finished containers through a blow molding process, often using the heat and pressure to shape the preform into desired container. So when we give heat and we blow air, so that will preform will be into the shape of some type of bottles. So this question is saying uses probes. So probes we used in option D, right? So option D is correct. Snorkel machines. Is this question clear?
Yes, ma'am.
Okay, clear to everyone?
Yes, ma'am.
Okay, thank you. Next question is about chilling injury. So which of the among the following statement is false with respect to chilling injury in fruits and vegetables? So chilling injury is a physiological defect of plants resulting in reduced quality by exposure to freezing temperatures. Second is low temperature induced membrane lipid transitions lead to membrane disintegration. Chilling injury results from the direct effect of reduced temperature on enzymes or indirect effect of membrane perturbations on intrinsic enzymes. D is lipid peroxidation may have a role in the development of irreversible injury during low temperature stress.
Option D.
Option
Option
D.
D is false. Okay.
Option A.
Anyone else? Option A is false.
Option A, madam.
Option A. So option A is false. Let's understand this question. So chilling injury, basically it is a psychological defect of plants and their products that results in reduced quality and the loss of product utilizing following exposure to low but not freezing temperature. So there will be temperature which is non-freezing. It may be less than 10, or it may be like, it depends, actually it varies on different fruit types. So for some fruits it can be around 10, and some fruit it can be around less than 5. If we decrease the temperature, chilling injury may occur. So since it is saying this option A, freezing temperature, which is not correct. It is low temperature, but not the freezing, which is 0 degree. No, not there. Greater than 0 degree, and maybe less than 5 degree or 10 degree. So the chilling injury was induced. So because of chilling injury, membrane lipid phase transitions happen, which further leads to the loss of membrane integrity and physiological dysfunction. It can also result from the direct effect of reduced temperature on enzymes, or it can be from indirect effect of membrane perturbations on intrinsic enzymes. It is also speculated that lipid peroxidation may have a role in the development of irreversible injury during low temperature stress. And there are different fruits like bananas and pineapple, which are having highly chilling sensitive. They are highly chilling injury sensitive fruits, and if we decrease temperature below 12 degrees Celsius, they will undergo chilling injury. So there can be different symptoms that our fruit is undergone chilling injury, that there can be pitting in the fruits, there can be browning because of the chilling injury, or the loss of the texture or the softening, or uneven ripening can also occur. So this is the biochemical mechanism of chilling injury. So what happens, since we discussed about this membrane integrity loss, so membrane, in membrane there is liquid crystals are there, lipid crystals are there. So because of the chilling stress, it will convert into solid gel. It will convert into solid gel. Then so it will convert into solid gel. Second is a free radical injury. So what happens, since the cell membrane is converted into solid gel, so there can be anything like increase in the cell membrane permeability. So because the cell membrane liquid crystals has converted into solid gels, and because of that there may be higher electrolyte leakage, and because of that cell may damage or lysis of the cell can happen. Second is there may be increase in the CO2 cytosolic levels. So what happens, because of that free fatty acids will be more, and because that will further oxidize, and fatty acids peroxides will form, and then it will lead to cell membrane oxidation, and finally cell damage or cell lysis. Third can be functional disturbance of membrane proteins can happen. Because of that there will be decreased photosynthesis and respiration, and active oxygen radicals may produce, and it can lead to the formation of the cell membrane oxidation, or maybe the fatty acids, they can peroxides, like the peroxide forms will form, and further the peroxide fatty acids peroxidation will also lead to cell membrane oxidation, and finally cell damage or lysis will happen. So there, in this diagram, you can see that in the banana, because of chilling injury, browning we can see, right? So in the second diagram, this is avocado. So in avocado, we can see there are pitting. So these are the locations where pitting we can see because of the chilling injury. And third is the cucumber. So this is cucumber. In the cucumber, there is water
Ma'am, your
spots
voice
or
is not audible.
Madam,
voice
it is
is not
audible.
okay. So can you please check it's audible to other people?
Yes, ma'am.
Audible, ma'am.
Okay, so we are continuing our discussion. So this is cucumber. Diagram is of cucumber, where without peeling we can see like this, and after peeling we can see there is a formation of such water soaked spots because of the chilling injury. So option A, which is false about chilling injury, rest options are correct because it is talking about freezing temperature, and in chilling injury we are greater than, we usually use greater than 0 degree Celsius. So is this question clear?
Yes, ma'am.
Yes, ma'am.
Yes,
Okay.
ma'am.
Okay, ma'am.
Okay, so let's move to next question, which is about numerical. So a food material is packaged into the LDPE packaging material, which has surface area 400 centimeter square, and thickness of the packaging material is 0.1 centimeter. Permeability coefficient is given as 4.18 into 10 to the power minus 8. Partial pressure difference of oxygen across the film is 0.21 atm. Calculate time in hours in which level of oxygen inside the packaging materials becomes 2.1 centimeter cube. It is a similar question as before. So please solve this question. So let's solve this question. We have the thickness of film given in the question as 0.1 centimeter. Surface area is 400 centimeter square. Partial pressure difference is 0.21 atm. Permeability of the packaging material is 4.18 into 10 to the power minus 8. So, and also the volume of the oxygen gas inside the package is given as, final volume is given as 2.1 into 2.1 centimeter cube, and we need to tell the shelf life of the food material or time we can keep the food material. So as we already discussed, that the permeability coefficient is equals to volume into thickness divided by area into time into difference in partial pressure. So time we need to calculate here. So shelf life of food material in time is equals to volume into thickness divided by permeability coefficient. It will come here in the denominator into area into difference in partial pressure del P. So shelf life of food material T is equals to Vx by Pm into A into delta P. So V, which is the volume of the oxygen inside the packaging material, is given as 2.1 centimeter cube. So we can keep 2.1 centimeter cube into thickness of the material is given, film material is given as 0.1. So into 0.1 divide by coefficient, permeability coefficient, which is 4.18 into 10 to the power minus 8. So 4.18 into 10 to the power minus 8 into area, which is 400, like this, into pressure difference, which is, sorry, 0.21 atm. So this is 0.21 atm. When we solve this part, we're going to get 59,808.61 seconds. So we can divide by 60 into 60 to convert it into hours. So it will be 16.6 hours. So option B is correct. Is this question clear?
Yes, ma'am.
Yes,
Yes,
ma'am.
ma'am.
Clear to all, right? So we are
Yes, ma'am.
moving to the next question. Yes. So what is the respiration quotient of the reaction? So this is the reaction given and we need to tell what is the respiration quotient in this reaction. So this is the easy one, right? What is respiration quotient? We just discussed in few slides back. Please,
Madam,
Zero
moles
point
of
anyone,
CO2 produced divided by moles of O2 consumed.
right?
0.7.
0.7. So
0.7.
both of you are, yes, both of you are correct. So the ratio of CO2 produced to O2 consumed is our respiration quotient. So RQ is equal to RCO2 divided by RO2. So in the CO2 it's 102 divided by O2 is 145. So 102 divided by 145 is 0.7. So option C is correct. So both of you are correct. Good job. So is it clear to all? Can we move to next question?
Yes, ma'am.
Okay, next is about a numerical. So kiwi fruits are stored in controlled atmospheric storage unit and producing ethylene at a rate of 0.1 microliter per kg per hour. If the required ethylene concentration in the inside the storage unit is 40 parts per billion ppb, so what will be the output capacity of ethylene decomposer for 2 tons of kiwi fruits with an efficiency ratio as 0.95? So this is very straightforward question. Directly it is based on the formula which is given in the last lecture video. So please try to solve this, which the lecture video about on a controlled atmosphere and modified atmospheric storage units. In that particular video, the one formula for calculating output capacity of ethylene decomposer is already given. So please try to see and calculate and let me know. Please solve. So have you seen the formula? Anyone? What is the formula from the last lecture video?
R into M by C into D.
Mm, correct. She's correct. So let's
Option
solve this.
A is correct.
Yes, yes. Option A is correct. So she has done a nice job. Everyone, please see the formula. Please put some efforts while I'm saying to see the formula. Please see in the video and solve the question. So nice work done. So let's solve for others. So it is given in the question that total quantity of kiwi fruits is 2 ton, which is 2000 kg. 2 into 1000 because it's in ton and we are converting into kgs. Ethylene production by the fruit is 0.1 microliter per kg per hour, which is given in the question, and required ethylene concentration is 40 ppb. So it is in parts per billion and we are converting parts per million. So we are multiplying with 10 to the power minus 3. Efficiency ratio is 0.95. So we need to calculate capacity of ethylene decomposer. So this is the formula I pasted from the video lecture. So the capacity of ethylene decomposer can be estimated by the formula F equals to R into M by C into D. So F is output of the decomposer, M is the total tonnage of the fruits, R is the ethylene production of the fruits, C is the required ethylene concentration, D is the efficiency ratio. So R, which is ethylene production. So ethylene production is 0.1. So 0.1. M is the total tonnage of the fruits, which is 2000 kg. So here into 2000 divide by C, which is required ethylene concentration in ppm. So 40 into 10 to the power minus 3 ppm, which is, and D is the efficiency ratio, which is 0.95 or 95%. So when, please can you mute? Thank you so much. So when we solve this, we will going to get around maybe 200 divided by 40 into 0.95. So here we can write is 0.2, right? So this we will go, so this we will get, and when we solve, we will going to get 5263.15 liter per hour. So when we want to convert it into meter cube per hour, so 1 liter is equals to 10 to the power minus 3 meter cube. So 5.26 into 5.26 meter cube per hour is the answer, which is A option, as correctly mentioned by her. Is it clear to all now?
Okay, ma'am.
Yes, ma'am.
Yes. So let's move to the next question. When there is no increase in the respiration rate of food material with changing the temperature, the value of Q10 is. So we know that we already discussed in our very first week lecture that Q10 value, which is temperature coefficient, is factor by which the rate of a reaction increases for every 10-degree rise in temperature. So formula is this. So Q10 is equals to K2 by K1, 10 divided by T2 minus T1. So K1 is the reaction rate at temperature 1 and K2 is the reaction rate at temperature 2. So this is the Q10 value. So now question is saying when there is no increase in respiration rate of the food material with changing the temperature. So what will be Q10 value? Please can anyone tell? Hint is also provided. So let's see. It is 1. So the temperature coefficient over 10 degrees Celsius interval describes how the respiration of a commodity changes as the temperature is raised or lowered. So every 10 degrees Celsius, what is the change? We denote it by Q10 value. So Q10 value are usually highest between 0 and 10 degree, which is 2.5 to 4, and then decrease to 1 with no increase in respiration as temperature near the tissue's thermal death limit. So here answer 1, because when there is no respiration rate, then with the changing in the temperature, then Q10 will be 1, and it is the condition where tissue is thermal death limit, it's near. Is it clear?
Yes, ma'am.
Yes, ma'am.
Yes. Okay. So let's solve next question. Orange juice is aseptically packaged in a laminated pouch and held at 25 degrees Celsius. The degree of browning as OD, which is optical density at 420 nanometer, measured on first day is OD value on the first day in the juice is 0.1. The Browning reaction, it is following zero order kinetic or zero order reaction. The rate constant for juice is 2.375 into 10 to the power minus 3 OD per day. Calculate the shelf life of juice in days. If the juice become unacceptable when the OD or the degree of browning exceeds 0.3 OD. So initial OD is 0.1 and final is 0.3. We need to calculate time. We need to calculate time or the shelf life. Rate constant K is given as 2.375 into 10 to the power minus 3. So apply zero order kinetics reaction. So what is zero order reaction? Please anyone? This is a very basic chemistry question. We have zero order, first order, second order, right?
Yes, ma'am.
OD final minus OD initial is equal to
Is equals to
change in OD.
change in OD final minus OD initial. Then I'm so sorry, I cannot, while I'm talking or while I'm on the slides, I cannot see chats. So I'm so sorry for that. So let's solve this question. Given is initial value of the degree of browning is at OD 0.1, final value is 0.3, rate constant is 2.375 into 10 to the power minus 3 OD per day, and we need to calculate the shelf life in days. So zero order reaction is the reaction where is defined as a typical type of the chemical reaction in which the rate of reaction is independent of the concentration of the reactants. That means the reaction proceeds at a constant rate regardless of how much reactant is present. So this is the plot where we can see that constant, like we can, this K, which is a slope, it is minus dA by dt. So we can see from this formula dt here means time is here, and this is the concentration difference divided by K. So T, which is shelf life, is equals to BE, which is BE minus B naught, which is the change in the OD divided by the K value, which is rate constant value. So BE, which is the final OD is 0.3, initial is 0.1 divided by K rate constant, which is 2.375 into 10 to the power minus 3. So in the numerator, it will be 0.2 divided by 2. This value 2.375 into 10 to the power minus 3. So the answer is 84.2 days. So option D is correct, 84.2. Is it clear to everyone?
Yes, ma'am.
Yes, ma'am.
Yes, ma'am.
Yes, ma'am.
So it's clear to everyone. We are moving to next question, which is also very simple question about edible coating material. So which of the following option indicates the correct flow line of preparation of edible coating material? So A is addition of glycerol, then addition of sodium alginate, which is a biopolymer, with continuous stirring, then heating in the water bath. B is saying heating in water bath and then addition of glycerol, and lastly addition of sodium alginate with continuous stirring. Option D is
Ma'am,
C
option
is, let me finish, I'm so sorry. Addition of sodium alginate with continuous stirring and then heating in the water bath and then addition of glycerol. And option D is addition of sodium alginate with continuous stirring, then addition of glycerol, and finally heating in the water bath. Please now put your opinion.
Ma'am, option C.
Option C.
Option C, ma'am.
Option C. Everyone.
Option C, ma'am.
So everyone is on option C. So let's see the solution, which is option C. You guys are correct. So the correct flow line of for the preparation of edible coating material is, first we take millipore water as a solvent. First we take any solvent which is mostly for biopolymers, mostly are water soluble. So we take millipore water and then we add biopolymer, which is sodium alginate, and then we do continue with the continuous stirring. We add sodium alginate. If we do not continuously stir, then because sodium alginate is very like is highly hydrophilic, so what it will do, it will form agglomerates and the clogging will be there, and it will not mix uniformly in the whole water or in the whole solvent. No uniform and effective mixing will be there. After that, heating in the water bath. So heating usually promotes uniform mixing, and if um, usually in sodium alginate there is no bond formation, but in sometimes in another biopolymers it may happen that there is a form or form, there is a formation of some kind of bond. But in the sodium alginate or other water soluble biopolymers, there can be formation of the hydrogen bond with the water, and it the reaction or the rate of the formation hydration will increase as we increase the heating. So there will be more rapid formation of the uniform and effective solution. Then there will be finally addition of glycerol. So glycerol is basically a plasticizer here. So what glycerol do is, or what plasticizers do is, there is a 3 hydroxyl group is there, which will form hydrogen bond with the hydroxyl groups. Hydroxyl group of the any polymer, biopolymer, which can be sodium alginate, starch, or cellulose, which effectively disrupt the existing hydrogen bond between polymeric chains, and there it can provide flexibility and ease of the processing. So if there is this chain of polymer, this chain of polymer, and it is bonded with hydrogen bond, so plasticizer will go inside and will form bond with this chain and this chain. Some plasticizer will form bond with the upper chain and some will form with the lower chain. So plasticizer will go inside the intact structure of the polymer, like this is the intact structure or the crystalline structure of the polymer. Plasticizer will go and it will go in the free space or increase the volume, and because of that the flexibility, because the plasticizer is forming bond now with the polymer chain, so flexibility will increase, and also it will be easy for process any polymer. So is this question clear to everyone?
Ma'am, what is millipore water?
This is just like distilled water is there. So one more upgraded form is millipore water. We usually use millipore water for the experiments, and it is the deionized form of the water. So there will be no ions like calcium, or the usually what we drink, the water we drink is usually having lots of minerals, and the uh, eh, like Ca plus and many minerals are there. So in millipore water, all these ions or the minerals, they will be taken out. So there will be no minerals. So because what happens, this if there is mineral in the millipore water, so like calcium, Ca plus 2. So what it will do? If there is polymer chain is there, so Ca2, it will form cross linking.
Okay.
So yeah, so that's why for experiments, just like we usually use millipore water or the distilled water.
Okay, ma'am. Thank you.
Okay. Can we move to next question?
Yes, ma'am.
Ma'am, can you please explain the uses of alginate and chitosan in like what kind of particular food groups we actually do a coat of this alginate and chitosan?
What is there? There is no as such food where only chitosan can be applied, or there are no as such food where sodium alginate is only used. Actually, this is very highly researched area, and this is like from very past, from wax coating. Like initially, what happens? There was the wax coating there. Eventually, it moved to edible coatings, like from the different biopolymers or the edible waxes also. So these from the the chitosan is very effective biopolymer, but it has religious concerns. Like what happens, the because it is extracted, the chitin is extracted from the exoskeleton of the insects, and mainly it is derived from the animal based sources. So it has religious concern. People from vegetarian, like the vegetarian people or people who are doing fasting, and the people who are only vegan, or there may be people which are allergic to chitosan, so they usually restrict the use of chitosan. Otherwise, chitosan it is having very good antibacterial, antifungal property and very good film forming property, and it was a one which like right now if you go and search on the Google, so you will find several like many researches on strawberries, on like whatnot. So chitosan is widely used on many fruits, mango, tomato, not even on the fruits, even on the vegetables. But sodium alginate, we consider it as a like a for vegetarian people or people who do not prefer animal based biopolymers. So slowly people, because of that gap, uh, people started working on the biopolymers which are extracted from mainly like from plant sources, maybe sodium alginate, or different gums are there, xanthan gum are there. So people started exploring those options. So sodium alginate is from, basically it's from the algae, which is also considered as a plant based. So uh, like it is used, and also like pectin. Yeah, one is pectin. So pectin is also very much used. So there's no uh, as such thing that this kind of uh, biopolymer can be applied to such particular fruits or vegetables. But there are like two different groups of people, the vegetarian and non-vegetarian. So based on that, people started exploring more plant-based biopolymers. So sodium alginate is one of them, like pectin is another. So there are different research. If you see for sodium alginate also, they can be they are also like in the research phase. They are applying, some researchers are applying on the strawberry, some are on oranges, mandarins. So is
Actually,
this
there is a
clear?
question map, assignment 9, a week 9 assignment. Like there is a matching question for alginate, chitosan, wox, and starch. As
Hmm.
chitosan only explained, but what kind of particular food group it is there, it is not mentioned there. But the options are like coating for confectionaries, coating for seafood, coatings for fruits and vegetables, coating films
Mm-mm.
for fish produce, like that. There is like in notes, it is not much detail it is there, so we
Mm-mm.
shows
Okay,
once.
okay. So you can see that for marine products or for maybe sea products, you can try chitogen or like that. So mainly, if you see even in the Google Scholar, you find the different review papers or the research papers. So it's like not fixed that this kind of coating will be applied to this. Even there are no as such food regulation in the FSSAI about these coating materials. Uh, so like difficult to say which food material is we can apply different coating.
Then how will
So
do match that question, ma'am? Because
so
it's there.
most relevant, you what you do is most relevant uh, you can find the most relevant, like for seafoods and like that chitosan can be applied, right, like that. What are
Okay, ma'am.
different polymers, what are different polymers there mentioned in the assignment?
Polymers. No, ma'am. This is the question for inedible coating. Like
Mm-hmm.
alginate is used for what kind of coating? Whether it is for confectionaries or for seafood or for fruits and
Okay,
vegetables or films for fish produce.
okay. So I think mostly it will be for fruits and vegetables. So is there any case study provided in the lecture or video about sodium alginate?
No, ma'am, that's what only a chitosan is explained, but it is not explained like for what kind of food group it is used. Rather,
Mm-hmm.
all things are not mentioned, so we are facing issues while solving this question.
Okay, okay. So I think mostly for fruits and vegetables you can say, but actually it's difficult to
Would
say,
be
to
for
be very
fruits and
honest.
vegetables. No? For walks, it is fruits and vegetables for walks, no?
I'm sorry, I'm not able to understand.
Actually, ma'am, other option is also there, no? Walks, like coating for fruits and vegetable. If we'll do answer is walks, then for alginate or chitosan, what will do?
Oh, match, right? Yeah. So
Ma'am,
chitosan can be mainly for meat kind of products.
okay.
Hmm.
Starch could be.
Uh, Starch. What are the options?
Uh, Ma'am, it is better if you will go for assignment 9 question. It is there.
Okay,
Question.
maybe yeah, maybe we can discuss at the end because we have other questions to solve for this. Uh,
Sure. Ma'am.
yeah, so we can go at the end. Uh, yeah, but that is better, I think, because we are solving today's questions.
Sure, ma'am.
Okay, thank you. So next question we need to finish because time is running. Yes, we need to finish this. So the cold storage measures the cold storage is their facility and it has so it has a dimension of this is a like a room or rectangular room which has a dimension of 6 into 6 into 3 meters, which is length, width and height on the inside, and is constructed of bricks laid in cement mortar. So what the wall thickness is 40 centimeter and 10 centimeter is the isolation, cork isolation on the inside of the four walls. The cement plaster is 1 centimeter thick. The heat transfer coefficient for ceiling is 20% more than that of the walls. The outside temperature is 30 degrees Celsius and the inside is maintained at 5 degrees Celsius. Calculate the total heat load in kilocalories per day due to conduction. So here the main mode is conduction, and the thermal conductivity of the bricks, cork and cement plasters are given as 0.45, 0.025 and 0.25. So this is kind of a multi-layer wall setup, right? Like this in series, and conduction is going on. So uh, we need to apply Fourier's law here. So please, can anyone let me know what is Fourier's law? So let's solve this question because we don't have much time now. So there is given the thickness of the brick wall, which is 0.4 meter. Thickness of the cork insulation is given as 10 centimeter, which is 0.1 meter. So thickness of the plaster, which is given as 1 centimeter thick, so it is 0.1 meter. Thermal conductivity of bricks is 0.45 given in the question, and then cork insulation it's 0.025, and for cement plaster it's 0.25, right? And the inside temperature and the outside temperature are given as 5 and 30 degree respectively. So this is our whole setup. Like this, we have different layers of cork, brick and cement, plaster, and it is in series. The Fourier's law, it says that the rate of heat transfer is directly proportional to wall surface area into temperature difference divided by wall thickness. Calculation of overall heat transfer coefficient, we need to calculate in a multi-layer heat conduction system, which is this is the multi-layer heat conduction system. In this we need to calculate overall heat transfer coefficient first to apply this formula, because in this type of setup, rate of heat transfer Q is equals to temperature difference divided by overall heat transfer coefficient, but we need to calculate overall heat transfer coefficient because it's a rectangular composite wall system in series. So this, and here is the A is the area. So 1 upon U is equals to x1 by k1 plus x2 by k2 plus x3 by k3 in a rectangular composite wall system, in that to be in series. So this is the formula for overall heat transfer coefficient U. And x1 here is the particular thickness of different layers, and k is the thermal conductivity. So 1 upon U is equals to x1. x1 is, which is thickness of the brick layer. First is brick layer. So 0.4 meter. So here is a 0.4 meter, and its thermal conductivity is 0.45. So divide by 0.45 plus x2 is thickness of cork insulation, which is 0.1. So this is 0.1 as thickness, divide by thermal conductivity, which is 0.025 plus x3, which is this. Hello, am I audible?
Yes, ma'am.
Yes, ma'am.
Okay.
Yes, ma'am.
Okay. Thank you. So thickness of cement plaster is 0.01 divided by conductivity of thermal of this cement plaster is 0.25. So 0.4 divided by 0.45 is equals to 0.89. Then second is the 0.1 divided by 0.025 is fourth. When we divide it, we'll get 4. And then 0.01 divided by 0.25 is 0.04. When we sum them, it will be 4.9. So, and this is 1 upon U value. So, U will be 1 upon this value, right? 4.93. So, it will be 0.203. Now, this is kind of a, this is our unit, CA or MA unit. This is there. So it is given dimension 6, 6, 3, 6 into 6 into 3. So this is the dimension 6 into 6 into 3 meters, right? So we need to calculate the heat transfer which is happening through the 4 walls of a room and from the ceiling. So 4 walls means this wall, right? So it is given 6 into 6 into 3. So for calculating area, this is like length into breadth into height. Height of the room is 3, right? So total wall surface area is equal to 4 walls into width into height, which is 4 into 6 into 3. So because we will, for wall we will take this rectangular, right? So this is 6 and this is height is 3. So 6 into 3. So 4 into 6 into 3 is 72 meter square. So from this we can calculate rate of heat transfer through the wall, which is the rate Q1 is the rate of heat transfer through the wall, divide by we divide by time because it is rate, equals to U, which is we calculated overall heat transfer coefficient into area of the wall into T1 minus T2, which is the thermal gradient. So Q1 is equals to, this Q1 will be, because we need to just calculate heat transfer. So it is heat transfer, not rate now. So heat transfer will be Q1 equals to UA into this thermal gradient into, now T will come this side, right, in multiplication, which is time. So Q1 is equals to 0.203, which is we calculated as overall heat transfer coefficient into area of the 4 walls, which is 72, into thermal gradient, which is the outlet temperature minus inlet outside temperature minus inside temperature, which is 30 minus 5, into time, which is 24, because we need to calculate in per day. So because this is hour, right? H means hour. So already we have hour and we need to calculate into day. So we are multiplying with 24. Okay. So 30 minus 25 is 25. So 25 into 24 into 72 into 0.203 will be 8769.6 kilocalorie per day. Heat transfer now we need to calculate through sealing. So Q2 is equals to UA into T2 minus T1 into, sorry, this is T, which is time. So yeah, so U we already calculated as 0.203, and but it is in the question given as that heat transfer coefficient for sealing is 20% more than that of walls. So we need so we are like we have to increase by 20%. So 1 plus 20 divide by 100. So it will be 0.2. 1 plus 0.2 will be 1 plus 0.2 will be 1.2. So 0.203 into 1.2, then area will be length. Now this ceiling is this part. So 6 into 6, right? Length into width. So 6 into 6 into temperature gradient, which is 30 minus 5, into time is 24, because we are calculating in days and we have this in hours. So 0.203 into 1.2 into 36 into 25 into 24 will be 5261.76 kilocalories per day. Now we need to calculate total heat of conduction, which is through the ceiling Q1, Q2 plus through the 4 walls, which is Q1. So this we have calculated 8769.6 plus this value, which is through the ceiling 5261.76, equals to this 14,031.36 kilocalories per day. Is this question clear? We have option A as correct. Is this question clear to everyone?
Yes, ma'am.
Yes for all?
Yes, ma'am.
Okay,
Yes,
thank
ma'am.
you. Thank you so much for letting me know. So let's solve the last question for today, which is which of the following satisfies the condition of ultra low oxygen storage? Anyone? 1 to 1.5% CO2 or O2 or 0.5 to 1% O2, and in other option it's like 4 to 5% CO2 or 2 to 3% CO2. So in ultra low oxygen storage, what is the amount of oxygen we use?
I think option C, ma'am.
Option C. Anyone else? So option D is correct. We have 2 to 3% CO2, not 4 to 5%. So in ultra low oxygen is a kind of controlled atmospheric storage technique where there is a reduction in the O2 partial pressure to the minimum tolerance level of the fluid, which is hypoxia condition. So there will be oxygen very low. So in ultra low oxygen storage, it has the oxygen level close to the minimum level required for the maintenance of plant tissues. Lower level will induce disorders such as browning and tissue narcosis, that is at 1 to 2 degrees Celsius with a level of 0.1% O2 and 2 to 3% CO2, which is option D. So we maintain such condition of very low oxygen. Ultra low oxygen decreases the respiration rate and ethylene production, which delays fluid ripening and reduces the oxidative stress with many psychological disorders. So when we lower the oxygen partial pressure, uh, then below the oxygen limit, then it will result in the anaerobic reactions like the fermentation reaction. So oxidative psychological effects can also, like we have the both the positive and the negative effects during this time. So positive psychological effects we do in the like uh, until when lower until the positive psychological effects will be there and very less fermentative or very less anaerobic reaction takes place. So option D is correct. So today we discussed about respiration and ripening, factors affecting respiration and ripening, and then the physiochemical changes associated with the ripening and the modified atmospheric storage, like the principle of modified atmosphere and the methods for modifying the gases composition. Then we discussed about modified storage, atmospheric storage model, and then we discussed about active packaging. We discussed about its principle and mechanism. We discussed about edible coating mainly. We discussed about panning, and then we discussed about CA and MA storage units and about their design, and we solved numerical about it. In the next week, we will be discussing about grain storage, then ozonation of food grains, hyperspectral imaging for quality analysis of food grains, non-destructive methods for analysis of grain quality, and last is the detection of spoilage in grains during using biosensors. So this will be discussed in the next week. So now let's discuss about the queries.
Thank you, ma'am.
Yes, thank you. If you want to leave, you can leave. If you want to discuss queries,
you,
now
ma'am.
we can discuss queries. Thank you.
Thank you, ma'am.
Thank you so
Now
much.
we can
Thank you, ma'am.
Thank
that
you so
question.
much. Rate of
Thank you, ma'am.
reaction independent. Thank you so much. Linda. Oh, the question is posted maybe.
Yeah, ma'am. Somebody already texted in the
Yeah,
chat
yeah.
box.
But in the exam, only calculation will be there.
No, no.
Both
To
kind of. No. I was just reading all the comments.
okay, ma'am.
Yeah.
Fine.
So let me see this question now. Alginate, chitosan, wax, starch. Coating films for fresh produce, coating for fruits and vegetables, coating for seafoods, and coating for confectionaries. So it's like quite confusing actually, because if we talk about like if a professor has discussed about this, we can tell. But usually, what if you see in the Google also, what happens? There are like this alginate coatings. They are applying for fruits also, for more like seafoods also. So mainly like, and commercially as such, no coating is FSSAI approved, or there's no regulations for the coatings. So I think I also need to see this question. And are you all there on the on the discussion forum? So today only, just after this class, maybe in 1 hour or 2 hours, I will post. I can post this solution on the, maybe on the chat box of Google, of the discussion forum, because actually this is quite confusing.
Similarly, ma'am, the 9 number question also, like the same kind, because it is not explained during the class.
Okay, not explained in the class. Question number 9. What is
Yeah,
question number 9?
that the same person has already shared the same question also, 9 number.
Okay, in the chat box.
Yes, ma'am.
Chat box. I'm already accessing it. Yeah. So where is the next question? Maybe this
Another page, a first page.
first page. This one.
Yes, ma'am.
This one is also the same question. Okay, ninth question. Match
Yes, it's
edible coating materials given in this with the characteristic. Okay, call chitosan, wax, starch, alginate, high water permeability, poor water resistance, good barrier for oxygen but low for. This question is possible to answer actually. If you uh, see, wax coating is mostly like forms a thick layer, and even people will not like to have wax.
yeah.
So there we can see that there are now, from you see, which are hydrophilic polymer, which are hydrophobic polymers, and based on that we can tell, right? So I cannot tell you the exact answer, but it is, I am saying this is possible to
Yeah,
answer.
yeah. Don't tell us, ma'am. It's okay.
Yeah,
This
yeah. I
like see in chitosan it is given high water permeability that is given to us during the class, but in Google something else is showing for chitosan.
So, since other polymers, if you see starch and alginate, they are water soluble. So, they will not give high permeability to water. So, you can say chitosan can be probable answer. I'm not saying it's the correct answer or not, but I'm just giving you some hint. So, you
Starch is
see
also
the
high
property.
water
Yeah,
permeability,
so
ma'am.
it is high. Starch is water soluble, right?
So it has also high water permeability, right?
Water permeability means what? Permeability means the like
It
starch
allows
what it
water
will
to
do. It will
penetrate.
take water. Yeah, it will take water and it will swell. So if there is a coating or a film kind of thing, so what it will do with water? Will it be allowing water to pass through it? What it will do? Permeability means
It is permeable. It is allowing.
transmission. Yeah, transmission of the water or permeability is water vapor transmission. Transmission is it is allowing water to transmit, right? And same is the with the permeability, right? That how much it is allowing water. So based on the property, you see the property, you will get to know. I think this can be answerable. Yes. So
Okay, ma'am.
only
Well,
questions. Yeah, just see the property of different polymers. It's easy to answer, not very difficult. Two options I already discussed with you, and only confusion is with alginate and starch. Even like wax is lipid, so
poor
what it can
water
offer?
resistant in the sense it is also water permeable, right? Poor water resistant, it means what? It is water permeable as well.
So you see water permeability definition, and is it resisting or is it passing through? Then you will easily understand. Good good barrier to oxygen, but for not for like moisture. So it will be something like related. See options. See the option. You will easily be fine because wax cannot be like low for moisture, right? It will be good barrier to moisture because it is a lipid. So just see the property. Just see. There are different three kind of biopolymers: polysaccharides based, protein based, lipid based. Characterize polymers based on that, and then you will based on the property you will able to find the answer.
Yes, ma'am. We'll try.
So yeah, because I cannot just say the answer because this is correct
It's okay.
question. There's no as such confusion in this question, but yeah, I admit that this question is quite confusing, and I will help you in this question.
Ma'am.
Yes.
Yes, ma'am. In assignment 8, question number 15, the answer is given, but it is showing wrong. Question
Assignment.
number 8. 8, question number 15.
Ma'am, I have
First
written in chat
assignment.
box.
Okay, let me check. 15 in top down, which one of the method is not used to generate nanostructure? We discussed, right? This kind of question.
Yes, ma'am, that is there, but what is taught in the class, that
Hmm.
we only did. The answer is written something else, and we didn't got the marks from that.
So that is me that what I discussed is correct. So oh, you need to just make a message that this is
Already so
this
many,
answer is
already
yeah.
so many people texted in the discussion forum that this has been taught and this is the
Mhm.
answer. Please make a re-evaluation on that. Like so many people did.
So so it will be re-evaluated. No worries on that.
Madam, with regard to the assignments, like, will we get any extended time because I enrolled for the course little late?
Actually, there is no extension of the time as already mentioned in the discussion forum. Are you a part of that?
In discussion forum, no, I am not in the.
I provided the link already in the previous lectures. I provided already the link. I think let me check in the chat. Mm,
No, can we do the assignment now and submit, or what is the procedure?
no, actually, it's like out of 12 assignments, I think there are 8 assignments. If you do, that is like good to go.
Okay. No, but now time is there because I am not sure because we enrolled late.
Yeah, but the assignments whose deadline already closed, you cannot do that,
So,
right? That
so
is
we
what I'm
can't.
saying.
No,
So,
that means we can't take up the exam now.
no, you cannot do assignment if the deadline is over.
No, but we are paid already. They extended the date for payment.
So you need to particularly ask them. I I think this is the group Google. I think this is only right discussion forum. I am posting the link here for discussion forum. If nobody, like if someone is left right now, can join. But there is like the assignment you need to actually discuss with them only because they
No,
have
we are
only the
not getting any.
control.
We are not getting anybody's number. Whom to contact?
Uh, can you discuss on this chat discussion forum? See, out of 12 assignments, best 8 will be considered for marking, but you have not attempted even 8 because it's just last 3 to 4 to 5 assignments left. So you must discuss with them because 8 is like you must do 8 at least.
No, but when the exam time fees is extended, why they didn't extend the
But I
assignment?
do, I cannot say, right, because I have no control on this because this is the administrative thing. I am not the administrative person, the right person to discuss with this query. So that's why I'm recommending you. Please, can you go through the chat box, this discussion forum, and let them know your query or your problem?
Okay.
Yeah, because I'm not really the person of from administration. I cannot help you. I'm so sorry that I cannot help you in this.
Okay, fine, no problem. Because that was been extended and we paid the fees, and now if they don't
Yeah,
allow, then it will become waste of fees.
yeah, correct for you. You are absolutely correct.
So
You
if they're
must
not
discuss with
allowed
them.
us to pay the fees, then it's fine, no problem. But they allowed us to pay the fees, and now if they tell assignment is not accepted, then it,
Yeah,
it is not a
y-
correct way.
yeah. Actually, I admit your problem. So this is the chat box, the discussion forum. I think this is the link I provided you right now. Please join and put your query. This is the, yeah, I'm putting it again. Please raise your query here because here administrative people are also connected. So they will help you with your problem. So can is this today's session okay for everyone, or you
Yes,
people
ma'am.
have yeah?
Yeah. Yes, ma'am.
So can we close today's discussion session, today's interaction session?
Yes, ma'am. Thank you
Yes,
so much.
Yes, ma'am. Thank you,
Okay.
ma'am.
So
ma'am.
I will be discussing that to you in chat box about your polymers and the applications. Okay.
Ma'am, I have a query.
Yes, please.
Ma'am, in the first numerical you have solved loss of water vapor.
Today's lecture.
Yes, ma'am. Yes, ma'am. Today's lecture.
Okay, let me see. Okay, this one.
I'm not able to see
Okay,
the
I'm
numerical
so
one.
sorry. I'm so sorry. I have unshared the screen. So can you see the
And
screen
in
now?
this, yes, ma'am. The next part, loss of water vapor.
Yes, please.
In this, why it is written as 1 gram equals to 1000 kg?
Because
And why we have
see, this is in 1.6 into 10 to the power minus 9 kg mole. This is kg mole, right? We need to convert it into the, this is, first it is in moles and second it is in kg. So number of moles into molar mass, we will do this, but we'll going to get in kgs, like 10 to the power 3. So we are multiplying with 1000 as well, and then we'll going to get grams.
Okay, no, that's okay. Like we are converting from kg to gram, no? Like 1 kg is
Yes,
equal to
yes, yes,
1000 grams should be written.
yes.
Yeah.
Oh, one.
Yeah. Okay, okay, I
Oh,
get that.
that's a. I'm
Okay.
so sorry. That's my mistake. Typing error. It's just it
That's
just
okay, ma'am.
got
This
out of my
just
mind.
created. I got confused because of this. That's
I
why
am
I
so
asked.
sorry. I
That's
am so
okay.
sorry. You are confused because of me. I am so sorry.
That's
Just
okay,
I
ma'am.
am correcting. 1000. Oh, sorry.
O-
1000
1
kgs.
kg equals to 1000 grams.
Yeah, yeah, yeah. I am just
Yeah. Okay,
1
ma'am.
kg,
Thank you.
1000 gram. Okay. I'm so
Thank
sorry
you.
if you are confused because of me. I'm so sorry. Okay.
Thank you.
So
Thank you, ma'am.
thank you for joining today. Let's just end today's session. Please try to come on the next Sunday also. Thank you so much for joining. Now you can leave the meeting.
Thank you, ma'am.
Thank you so much.